The following video is a tutorial of the thawing and plating process for Novabiosis cryoplateable human hepatocytes. For this process, you will need Novabiosis cryoplateable human hepatocyte vials, Novabiosis thaw medium, Novabiosis plating medium, Novabiosis maintenance medium, collagen 1 coated cell culture plates, a 50 ml centrifuge tube, pipette tips, 0.4% trypan blue solution, an optional overlay matrix. We recommend Corning Matrigel, serological pipettes. You'll also need a water bath that is warmed to 37 degrees Celsius, a centrifuge with the capacity to spin 50 ml centrifuge tubes set to 4 degrees Celsius, a pipette aid, a biological safety cabinet, and a 37 degree 5% CO2 incubator. First, you will need to complete your Novabiosis plating media. Novabiosis recommends adding 50 microliters of 50 mg per ml gentamicin to every 250 ml of plating media. We currently offer two types of thaw media, 7001 and 7001P. 7001P featured in this video needs to be warmed, 7001 does not. We recommend warming the medias for at least 30 minutes prior to use. First, retrieve the Novabiosis cryoplateable hepatocytes from the dewar or liquid nitrogen storage. Immerse the vial up to the cap in a 37 degree water bath. Be careful not to completely submerge the cap. Thaw until ice pellet is completely melted. It will take approximately 90 to 120 seconds. The vial of hepatocytes is complete when the ice spindle looks as shown. Remove the vial from the water bath. Spray with 70% IPA or wipe down with a 70% IPA wipe and transfer to the biological safety cabinet. Pour the contents of the vial into the 50 ml centrifuge tube containing the Novabiosis thaw medium. Remove 1 ml of the thaw medium and use to rinse out the contents of the hepatocyte vial. Ensure the tube's cap is tightened and invert the tube gently 3 to 4 times to ensure resuspension of the hepatocytes. Centrifuge the tube at 100 Gs for 8 minutes. Remove the tube from the centrifuge and return to the BSC. Discard the supernatant on top of the cell pellet. Resuspend the cell pellet in 1 or 2 ml of 37 degree Novabiosis plating medium. Gently shake the tube to ensure complete resuspension of the hepatocytes. Calculate the total cell count and cell viability using the trypan blue exclusion method. In order to calculate the total cell count and cell viability using the trypan blue exclusion method, to achieve a 10x solution, combine 700 microliters of Novabiosis plating medium with 200 microliters of 0.4% trypan blue with 100 microliters of the hepatocyte cell suspension. If you desire a different dilution, the volumes can be adjusted. Cell viability is calculated using the following formula: live cells divided by live and dead cells multiplied by 100%. Cell yield per mil is calculated using the following formula. You take the total live cells, divide by the number of squares counted, multiply all of that by 10,000, which is the hemacytometer factor, and multiply that by the dilution factor. To determine total cell yield, take the previous calculation and multiply by the volume of the cell suspension. 100 microliters of cell suspension was removed and added to a 10x dilution factor trypan tube. 10 microliters of the cell suspension trypan mixture is being added to the hemacytometer to be counted. Dilute the hepatocyte suspension according to the recommended seeding density found on the Novabiosis client plating instructions. Dilute the hepatocyte suspension according to the recommended seeding density from the chart below using Novabiosis plating medium. The chart below represents general recommendations. See the certificate of analysis for lot-specific recommendations of seeding volume and feeding volume. After adding the appropriate amount of diluent, gently resuspend the cells in the tube. Add the cells to the desired plate format. After the hepatocytes are added to the plate, visually inspect cell seeding density. If cells appear too dilute or too concentrated, adjust density as necessary. An appropriate seeding density is shown here. After inspecting cell seeding density, take cells to the incubator. Before placing inside the incubator, shake the plates in a north to south, east to west motion gently. As mentioned, gently shake the plates in a north, south, east, west direction. Do not use circular or figure 8 motions. Gently place the plates into the 37-degree 5% CO2 incubator. For the first 2 to 4 hours post-seeding, remove the plates from the incubator every 20 to 30 minutes and shake gently in the north to south, east to west direction before returning to the incubator. After these 2 to 4 hours, replace the media with fresh, warmed Novabiosis plating medium. Gently place the plates back into the 37-degree 5% CO2 incubator. 2 to 4 hours after this first media change with the plating media, replace the media with fresh warmed Novabiosis maintenance media. If you are using an overlay such as Matrigel, replace with 4° C Novabiosis maintenance media mixed with the overlay mixture. Gently place the plates back into the incubator until use. Refresh the maintenance media every 24 hours. This concludes the Novabiosis tutorial video on thawing and plating our cryoplateable human hepatocytes. If you have any questions, feel free to contact us. Visit us on our website, www.novabiosis.com.